The second DNA reaction is gene expression. Gene expression consists of two steps: transcription and translation. When cells go through transcription, they use DNA molecule as a template to make a copy of that DNA molecule in form of messenger RNA. And then they use messenger RNA in translation to make a protein. Let's start with transcription. As I said, when cells go through transcription, they use DNA molecule as a template to make a copy of that DNA molecule. So that means that transcription as a process is very similar to replication. But there are certain differences as well. So let's talk about those differences. The first one. The end product of transcription is RNA molecule, and the RNA molecule is much smaller molecule than DNA, and that means that when cells go through transcription, they do not copy the whole DNA molecule. They copy only one gene that contains information how to make a protein. Now let's talk about structure of RNA molecule. Same as a DNA molecule, RNA molecule is composed of three parts. It is a sugar, in this case ribose, phosphate groups that link those sugars together, and four nucleoside bases. Those are adenine, guanine, cytosine, and instead of thymine, RNA molecule has a uracil. So A, G, C, U. To explain a transcription as a process and its function, a lot of sources compare transcription to the library. So a library is composed of books, and each book contains certain information that we might need. For example, one book explains you how to fix your car, another one explains you how to make a pie, and another one explains you how to build up a house. Same as a DNA molecule. It's composed of genes, and genes carry information necessary for each particular cell. Let's say one gene explains cell how to make a protein. So when cells go through transcription, they read that information, and then when they go through translation, they go ahead and make a protein. So let's talk about how cells read those genes. Let's say we need to build up a house. We go to the library, we pick the book that explains us how to make a house, to build up a house, and then we make a copy of that book. This is what we call blueprint. Cells do pretty much the same thing. They go to DNA molecule, choose gene that explains how to make proteins, and make a copy of that particular gene in form of messenger RNA. This messenger RNA or blueprint is going to be used in translation to make a protein. So let's get back to transcription. Transcription begins when on certain area of DNA molecule bubble is formed. Within that bubble, two strands of DNA molecule will be separated and bases will be exposed. Now, since a copy of this DNA molecule will be RNA molecule, which is a single-stranded molecule, it means only one strand of double-stranded DNA will be used as a template. As I said, when two strands of DNA molecule are separated, transcription begins. And it means a complementary copy of RNA molecule will be formed. So what is the name of enzyme that will make that complementary copy of DNA molecule? The name of enzyme is RNA polymerase. If you look at this picture, you'll see this is our RNA complementary copy. It's almost made. And you also can see that RNA polymerase also moves from 5' to 3' direction when it makes RNA complementary copy. And also, if you look closer, you will be able to see that on RNA molecule, uracil is complementary to adenine. And then transcription continues until RNA polymerase reaches what we call termination sequence, means end of a gene. When RNA polymerase reaches termination sequence, end of the gene, then it means transcription is completed. RNA will be released and DNA molecule will be closed back into double-stranded DNA. After the genetic information is passed from DNA to RNA by transcription, it is used to make a protein by translation. During translation, amino acids are linked together in a proper order to make specific protein, which is a polymer or macromolecule. The name translation refers to translation of information from the language of nucleic acid into the language of protein. Before we start talking about translation, let's talk about RNA molecules. There are 3 types of RNA molecules, and all of them are used in translation. So the first one is messenger RNA or mRNA. We already know that the messenger RNA is actually a complementary copy of DNA molecule. It is a blueprint for translation, directory. The next one is called transfer RNA or tRNA. tRNA is actually a translator between RNA and protein. And the last type is called rRNA or ribosomal RNA. It is a part of ribosome, and as you remember, the function of ribosomes is to make a protein. The end product of translation is a protein. So let's start with the structure of proteins first. The protein is a polymer or macromolecules. And like all polymers, it's made of monomers, amino acids. So let's talk about structure of amino acids next. Each amino acid is made of 3 bases. This is what we call codon, 3 nucleoside bases that code for 1 amino acid. For example, let's look at this chart. Let's look at this amino acid called tyrosine. So codon for this tyrosine is U, the first base, A, the second base, and U, the third base. But if you look below this tyrosine, there is another tyrosine molecule here, and the codon for this one is going to be U, the first base, A, the second base, and C, the third base. That means we have 2 codons, UAU and UAC, which encodes the same amino acid tyrosine. So how is it possible? Yes, it is possible. Some amino acids have more than 2 codons and all of them encode the same amino acid. For example, let's look at this amino acid leucine. There are 1, 2, 3, 4, 5, 6 codons, 6 different codons that encode just one amino acid leucine. This is what we call redundancy. Redundancy means we have more than one codon for each amino acid. So why does it happen? Because out of 4 different nucleoside bases, there are 64 possible combinations can be made for only 20 amino acids. This is the only number of amino acids that cells require, including human cells. That's why genetic code is redundant. It has more than one codon for the same amino acid. Now let's look at this chart once again. Let's look at this amino acid which is called methionine. Methionine amino acid is also known as a start codon. And the codon of methionine is, as you see, A, U, G. So please remember, methionine is a actually start codon and the codon of methionine is AUG. When we go through translation, I explain you the function of this amino acid. And one more term I would like you to remember is nonsense codons. Let's look at this chart. There are 3 codons in this chart which are called stop or nonsense codons. As you see, each of those stop codons have codons. For example, this one codon is going to be UAA. For this one, it's going to be UAG. But neither of those stop codons encode any amino acids. That's why we call them nonsense or stop codons. And again, when we go through translation, I explain you their function. On this picture, you see a structure of T or transfer RNA. As I said, a tRNA, it is a translator in translation because it can recognize 2 languages. Language of amino acids and language of proteins. This end of tRNA is called anticodon, and it uses anticodon to read codons on the messenger RNA. And then that information will be passed on amino acid side, and on this side tRNA will make amino acid, which is encoded by specific codon. Please remember, each tRNA only can recognize one specific codon on messenger RNA. Alright, translation. So a protein produced by translation corresponds to a single gene. But most messenger RNA molecules are larger than a gene. Why? Because messenger RNAs have specific regions in their structure that play roles of signals for ribosome. Let's look at this picture. This is the messenger RNA with codons. This is the ribosome. And this specific region on messenger RNA is called Shine-Dalgarno sequence, or the ribosome binding site. This signal shows to ribosome where it is supposed to attach to messenger RNA. After ribosome attached to shine Dalgarno sequence on the surface of messenger RNA, it moves to start codon, the second signal for the ribosome. Start codon tells ribosome that a translation begins from this point. Let's look at this picture. It shows you the structure of ribosome. Each ribosome has two sites. This one is called A site or amino acid site, and this one on the left is called P site, means protein site. So everything begins when tRNA comes to the A or amino acid site and with its anticodon reads codon on the messenger RNA. Then it passes that information to amino acid site, where amino acid encoded by this codon will be made. The next step, that DNA with amino acid will be moved to the P site of ribosome, and it happens again and again and again, and those proteins will be connected, linked to each other as translation continues. So let's look at this picture. When ribosome moves along the messenger RNA, tRNAs read codons. Each codon on messenger RNA produce amino acids and then move them to the P side of a ribosome and link to each other. And ribosome continues move to the next codon, and next tRNA will read it and produces next amino acid. So when ribosome moves along messenger RNA, with the help of tRNAs, it reads one codon at the same time and produces one amino acid encoded by that codon. This process continues until ribosome reaches what we call nonsense or stop codon. This is the last signal for ribosome, which tells ribosome that translation is completed. Then ribosome will be removed from messenger RNA and the protein will be released. If cell needs to make another protein that is encoded by this messenger RNA, translation begins again. Then ribosome will attach to shine Dalgarno sequence, will move to start codon, translation begins from this point and continues until ribosome reaches stop or nonsense codon. What you have to remember about gene expression of bacterial cells is transcription and translation in bacteria can occur at the same time, and that explains why bacteria have higher metabolism. For example, in eukaryotic cells, in our cells, transcription occurs in nucleus, and then messenger RNA will be moved in cytoplasm, where ribosomes are located, and this is where translation occurs. Because bacteria are prokaryotes and they have no nucleus, then, as I said, transcription and translation can occur in the same place in cytoplasm at the same time. That's why bacteria grow and multiply faster and have higher metabolism. And to support that metabolism, they produce more ATPs, more energy containing molecules than eukaryotes.